have Shai Botanic and Jonathan Meuser, the founder. And to introduce Jonathan and the company, we have Alex Copeland again. Alex, go right ahead.
Thanks, Nick. Morning, everyone. So just a quick about with Kai Botanic. They were part of our 9th cohort at IndieBio right before COVID. And, we had a lot of things we liked about the company. I think it was one of the more unique companies in the fermentation space we've actually ever seen. And I think we've still been really big supporters and believers in the thesis of making complex plant products in kind of this really unique way that other modes of fermentation can't do. And one of the other things we've been seeing lately that kind of has reinforced our belief in the company is also when it comes to climate resilience and supply chain disruptions, the ability to still make a lot of products through precision plant fermentation as opposed to relying on the field when that gets more and more difficult. So yeah, I've been really excited to keep up with John and the team. So also looking forward to seeing his latest presentation. Thank you.
Thanks a lot, Alex. So shall we just jump right in, Nick?
Go right ahead. Take it over.
Okay. Okay, so we're Kai Botanic, the plant cell company. Thanks for being here. Let's see. Okay, so already today, 40% of the Earth's landmass is used for human agriculture. So at the current scale, ag is causing negative impacts that are unsustainable now. However, demand for ag products is always increasing and requires even more resources. So ag is even more unsustainable long-term. Fortunately, consumers are becoming more aware and making smarter purchasing decisions. So ultimately, we need a new way to grow plants. So at Kai Botanic, by solving social and environmental problems of farming with our technology, or making a plant product available that's currently not accessible, plant cell cultures are the future of ag, and a new white space in both fermentation and plant ingredients. So most biomanufacturing is currently done with bacteria, fungi, or animal cell cultures. But unlike other cell cultures, plant cell culture can naturally make complex plant ingredients. Production is currently limited because most plant compounds can't be made in microbes, even with synthetic biology. So on the other hand, plant cell cultures are like plants, but can grow 100 times faster than they grow in the field. Also, traditional ag can take years to scale, grow, and harvest the products. Moreover, often tiny parts of the plant or low concentrations in plant tissue are currently needed. Whereas with plant cell cultures, every cell is making the product, and in months, not years. So here's how we do it. Plants can be readily made into stem cell-like tissue to create actively growing cell suspensions. For example, the aloe vera plant shown to the left. The challenge is optimizing the conditions and selecting the cells that make the desired product. Plant cell cultures have already been used to scale a few products, like the anti-cancer drug Taxol. But advances in industrial technology to customize and improve other cell lines, like bacteria, yeast, algae, or fungi, haven't been extended to plant cells. At Kai Botanic, we're leveraging the high throughput cell selection and methods that our team has successfully used to engineer commercial algae and fungi to commercial plant cell cultures that we've tailored to produce high value products in what we're calling the plant cell foundry. Initially, the plant cell cultures don't grow well under unoptimized conditions and are these mixed populations of diverse cell types, like this early aloe cell culture that you see pictured on the left. By using robotics and high throughput approaches, we perform selections and screens to isolate the best plant cell lines and identify the optimal growing conditions. So aloe is the first example of a cell line in the conditions that we've optimized for commercial production. So an aloe leaf normally takes as long as a year to grow, but for us, just 1 liter of our optimized cell culture can produce up to as much as 1 aloe leaf per day. On the right is a 15 liters of aloe culture in those bioreactors that produce the 200 grams or so of aloe powder that's commonly used in cosmetics pictured in the foreground. This amount of cell culture is actually equivalent to the yield of interleaf powder from the 55 kilos of aloe leaf that's shown in that picture. So these tools that we're using to identify the perfect conditions and genetically optimize the cell lines opens the door to a much wider range of products from plant cell cultures than ever before.
So products derived from plants is a $250 billion market, which spans multiple verticals. High value plant compounds can be used in industries like food and beverage, like vanilla and hops, cosmetics and personal care products like our aloe and various fragrances, medicine and wellness, and general manufacturing purposes. We have a very rigorous process for selecting our use cases, which is particularly important here when we have almost unlimited paths to pursue. So we built a database called KaiBase, which details over 400 plants that have high market value properties. Within that database, we're specifically looking for plants that have supply chain challenges, like they're either difficult to grow or they're inconsistent depending on their growing conditions or the weather, or they require unsustainable growing or processing methods. And of course, we're also factoring in things like the commercial value, the current market size, and whether the plant's biology is appropriate for the cell culture process. When we have hypotheses, we validate these by talking to the industry. We want to find out from companies that currently use that plant or that ingredient what their pain points are, what performance they would want to see from ours, and what their desired price range is. When we feel confident that there's a market need for a particular ingredient and we can satisfy that need, then we put that cell line into production. Our current pipeline consists of the 4 products that you see on the screen here: aloe, which is our go-to-market product, citrus, vanilla, and iris, and we have a handful of others in the very early hypothesis stage. So how do we choose aloe as our initial product? First, it's a huge market on its own at $1.6 billion, and it's pretty expensive at anywhere from $200 a kilogram to over $1,000 a kilogram for the most premium product. Second, aloe vera, of course, is very well known for its healing properties. It actually has over 200 bioactive compounds that are beneficial to the body. But the plant itself grows pretty slowly, as John mentioned earlier, and it contains a toxin called aloin, which is banned in the EU and it has to be processed out before using. The processing is expensive, and more importantly, can also destroy the beneficial compounds that we're after. So our solution is the Botanic cellular aloe. Using our platform, we're able to grow aloe cells without aloin from the start, which creates the only naturally non-toxic full spectrum aloe available. So we've succeeded in producing Aloe 2.0, and we've also secured our first pilot customer for it, Freestyle Beauty, which is a clean label premium beauty brand. Freestyle is committed to purchasing 200 liters of our cellular aloe, and over the next 6 months, we'll be formulating it into a cream cleansing mask and a moisturizer. These products should hit the market in Q4 of this year, and they'll prominently feature Botanic cellular aloe as their #1 ingredient on the label. The ability to provide their customers with this most sustainable and purest form of aloe available is a major brand differentiator for Freestyle, and they're willing to pay a premium for it. But our cellular aloe isn't limited to specialty markets just like cosmetics or sustainably minded brands. We see this pilot with Freestyle as a stepping stone to let us gather data and hone our production process so that we can increase our production capability to over 1,000 liters. At that mass market scale, we should be able to bring the cost of our aloe down to $5 a kilogram, which is significantly less than that $200 a kilo current floor. As we scale and we develop additional products, then more verticals open up, including food and beverage and nutraceuticals. And we're already pursuing talks with many of those customers.
Thanks, Jen. So Botanic's management team was first made up of co-founder Robert Jinkerson and myself. We were in the same lab during our PhDs working on algal genetics. Sachin Jain recently joined and is bringing his extensive experience with high throughput strain development of fungi to apply to plants now. Jen Goggin, who you just heard from, is a founder of multiple successful food tech startups and our key industry advisor that we're working together with on commercialization and business development. Over the last 12 months, a lot has been accomplished. We can now grow cells at the pilot scale, unlocking pilot production and capacity for high throughput R&D as well. For our technical goals, we've demonstrated the 2 things we've set out to do: select cells with the desirable traits and optimize conditions for growing the cells and making the products we want. We're already on the path to commercializing aloe and citrus, with vanilla and iris already being optimized in cell culture. So far, we've raised $2 million in pre-seed notes and an additional $1 million in non-equity funding through partnerships and grants. We're currently raising a $4.5 million seed that's flexibly 50% subscribed, with a targeted close for May 15th to fund 18 months of runway until our Series A. Using our seed funding, we aim to achieve the following targets. For revenue, our goal is 10 million of combined sales, product development, partnerships, and grants by 2024. We also aim to customize new cell lines using the plant cell foundry faster, with a goal to reduce the timeline from 12 to 6 months or less. And in parallel, we're scaling production of aloe and citrus to 1,000 liters, as Jim mentioned, the next step in the Series A goal of selling aloe and citrus into the mass market, some of the partners that we're talking to now and potential customers. And the company's ultimate goal of putting a significant dent in traditional ag with plant cell culture. So thank you. Please reach out to me at john@kaibotanic.com if you'd like to help us change how we grow.
Wonderful. Thank you so much, John. And it was really a pleasure to hear from you again. We have a little bit of time for questions here. I know Howard always has some. We also have some big brains in the room here that I'd love to throw it out to. We fortunately are having a discussion on just this set of issues at our workshop next week, led by Ron Meuser. So really looking forward to that. I'm sure there'll be a lot of relevance there. Ron, do you have any questions that you'd like to throw out to begin?
Well, first of all, I love what you're doing. Colleagues and I have been talking about the untapped potential of plant cell culture since, God, 18 years ago when I was running biotech R&D at Dow. And we were looking at a product that we were thinking of growing in crops. And in order to get some early samples to do some toxicology, we said, well, let's grow it in plant cell culture and see if we can just get enough to do our toxicology studies. And much to our surprise, we found we could produce in cell culture more cheaply than growing GMO crops. Somehow in the big corporate world, that great insight just languished and never went anywhere. So I love what you're doing. I think there's tremendous potential here. Question, I guess my biggest question is the predictability of cell differentiation. Many of the products you're looking for are only going to be produced in, the very specialized cells in a flower or in pistils or stamens or petals or early buds or that sort of thing, which you may or may not be able to duplicate in culture. Is that just a hit or miss? You've got to just try a lot of samples to find one? Or are you mutating the cells to try to induce the pathways that you're looking for in undifferentiated culture cells? What's your experience been so far?
It sounds like you're very technically knowledgeable, and we should have some good conversations, including you can pull me aside and tell me what product this is that we should be making, since the patent should be expiring soon. Yeah, so you're absolutely right. That's very insightful. We are specifically, as Jen mentioned, part of our selection process from KaiBase is whether we think that tissue should be easier to emulate. So like the irones from the iris root are everywhere in the root. So if we can produce like a root-like cell, should we?
So you don't need specialty cells there. You got it. Yeah.
Right. So we kind of stay away from nectaries, honestly.
But vanilla now is different. That's at the other end of the spectrum, right? Because that's got to be from the floral tissues.
It's actually from the inner bean lamellae, placenta lamellae, technically. But there's some interesting traits about that. Again, we can talk about that we think that will make selection easier. And the bottom line is, it's kind of based on these knowledge that in complexly regulated systems like algae and fungi, you can take a natural physiological state like high photosynthetic efficiency or high oil production and so-called lock it in at the transcriptional level. And in plants, there's actually some new literature coming out that we're basing this on, where you have transcriptional control of complex pathways like 20 genes in the anthocyanin pathway or the phenylpropanoid pathway for some of the fragrance molecules in these like undifferentiated cell cultures. So there's hundreds of global regulators putatively in plants, these transcription factors with completely unknown function. And we know that there's tight control over these different tissues in plants. But you're absolutely right. It is somewhat hit or miss. And that's the idea that we have these short-term projects where we get some early data. And some of our partners, we talk about these milestone-based projects. And if it's proving to be looking impossible, then we can move on. Again, we have many potential targets.
Well, it's impressive you found a couple already, a couple of big markets already that could carry the company just with those.
That's the point we're trying to get across.
Okay. Well, thank you. No, this
perfect.
This is exciting.
Dang it.
Thanks, Ron. PJ, it sounds like you have a question. Go right ahead.
I do. Thanks, Jonathan, for the presentation. Maybe one question here. In the cell culture kind of scale up that you guys have seen so far, can you give us a feel for the speed and scale of growth compared to other mediums and their kind of speed and scale of growth, depending on the cell types you've worked with so far?
Yeah, so in general, the cells grow faster and faster if you stay on top of them and keep diluting them. And there's a general principle that it takes around 1 to 2 days generally. So like rice, BY2 cells, tobacco cells that have been grown in labs. And for us, it took us a little while to select those cells that are growing faster. Luckily, it's a form of evolution, right? So we're working with the organism. And so our allo cells divide in between like 1 to 2 days if we give them the right conditions, which is slower than Easter bacteria, but we're also using 40% of the biomass as our product. So in terms of our yields, we're not producing like Geltor's grams of collagen. We're producing like all of the cells are what people want. Not all, but all the soluble portion. So it changes the economics like a lot, depending on how much you're using.
Gotcha.
Great. All right, Howard, looks like we have time for one more question before I move on. Go ahead.
Hey, John, really interesting what you're doing. So my question is a little less technical, but more market focused. So, we've seen a lot of cellular production companies come through, but most of them are animal. By last count, I think I read somewhere that there may be 90 or more animal cellular production startups out there, but we don't see very many, or if any, plant ones. So my question is, why is that? Is what you're doing much more difficult than animal cellular production? And what are the differences?
Well, since we founded Botanic, there's been an increase. Well, since our funding at IndieBio, there's been an increase in the number of companies at 50% a year, year over year. So we do see more and more companies popping up. And, I think some of the early movers in the animal cell culture, made a big impact and got a lot of investment, and that's brought a lot of interest. I don't think that plant cell cultures are going to be harder necessarily, especially when it comes to selling it.
Okay, well, that's encouraging.